Jay,
Yes, sir.
so
Good evening, sir. Good evening, sir.
Ma'am, sir is not there.
Good evening, sir.
good evening, ma'am. Good evening.
Yeah, sir, you can continue, sir.
Yes, ma'am. Thank you. Yes. Okay, ma'am. Thank you, ma'am. Good evening, all of you, my dear students.
Good evening, sir.
Good evening, ma'am.
Good
Today
evening,
we-
sir.
good evening, Pa. Good evening. Today, what we are going to discuss about? Already saw it. So many things is that, isn't it? Virus. Last class, what we have seen, the part 3, human welfare, particularly the virus, isn't it? So remaining things we are going to discuss. Okay. Before that, I ask who stated the statement, the virus is a poison. Anybody else? Virus is a poison. The term was given by the name of the scientist called. Shall I give the options?
Yes, sir.
V.M. Stanley. Okay. Muller. Louis Pasteur.
So Pasteur, sir. Louis Pasteur.
Good. Very good. Okay. What are the two biomolecules present in the NEET virus? What are the two biomolecules? Do you know?
Sir, is it DNA and RNA?
Yes, one is simply called as, both are, both are collectively called as nucleic acid, isn't it? Okay.
Yes,
And
sir.
one more is there. One more. Will you please? Already we saw it. That one, a tobacco mosaic virus. It consists of two components. Is there? One is inside. What is that? And outside was it? What is that? I told. We discussed that things also. Will you please? No idea, isn't it? Okay. So any viruses, it consists of two parts. Is there? Just to imagine. So one is called as a nucleic acid, whether it may be a DNA and RNA or RNA. Sorry, it will not present in both. That is a question, assessment reasoning question or true or false question. They may ask whether the virus is contains both nucleic acid or DNA or RNA. Like that, they may ask the question. So many times they were asked. Okay, so that is important. Only presence of DNA or RNA. Okay, nucleic acid is the basic material. That is very, very important. Today, what we are going to discuss about, the one of the dangerous disease is called as measles. Okay, so measles is also one of the viral diseases. See here, what is the causative organisms? It is caused by an RNA, a paramyxovirus, and rubella virus. The measles is also caused by the RNA containing viruses. Okay, this is very, very important. Most of the cases very dangerous, such viruses. It has a nucleic acid, that is one is the RNA. It is very dangerous one. HIV is taken example. It is also a RNA viruses. And the similar to that, COVID also. Okay, these are all a very what is that? Very dangerous one. It is easily spread. Okay, that is the reasons. Paramyxovirus, rubella virus. So it is also called rubella disease. Measles also called as rubella diseases. Just to note it down. Already I told you. Remaining here also, measles means any one of the virus disease or bacterial disease. You must know which is the cause fatty organisms. Okay, then second one is what are the main symptom of that particular disease. Third one is how it will be transmit from the person to person. Okay, otherwise what are the vectors like that? What are vectors means what is the aerosol, airborne disease, sputum, okay, and also semi liquid format like that. And then the incubation period is also very, very important. And then the last one is called as diagnosis and then treatment. These are the subheading. You must note it down very carefully. Okay, here also the measles causes paramyxovirus and rubella virus. Okay, so that's why it is called as rubella diseases. It generally affects the children in the age group 3 to 5 years, only the below 5 years. Okay, so here, okay. Epidemiology. It is a highly contagious diseases and spread by droplet infection. Droplet, note it down. This is very, very important. How it will be spread from the affected person to the healthy person by droplet infections. It is also transmitted to the developing of fetus through the placenta. Suppose the mother during the pregnancy, what happened? It may be entered through the food or whatever it may be. Okay. The droplets enter inside the mothers. It may be travel through the blood and also finally it reaches the fetus, developing of fetus, particularly through the placenta. Otherwise called as umbilical, the connection between the mother and fetus, isn't it? Okay. So incubation period about 10 days. See here, after 10 days only you will get the symptoms of measles. Okay. Okay, go to the next one. See here. This is a structure similar to that of the COVID. There is a highly hook-like structure. This is nothing but the glycoprotein, and also the center there is a cover is there. That is also one of the protein molecule. Inside, what happened now? There is a nucleic acid. Which one is the nucleic acid? RNA as a nucleic acid. See that the paramyxovirus. This is the structure of paramyxovirus. The outermost covering. See here. How many types of protein here? See that. So this is a very complicated one because it's also different types of protein also present in the paramyxovirus. See the hemagglutinin neurotransaminidase. Okay, neuraminidase. It's one of the amino acid. Okay, if this is called as the green color, the hook-like structure is also one of the protein molecule. Hemagglutins. Okay. See the fusion protein. This one is called F protein, M protein, that is what called matrix protein, P protein, phosphoprotein, N protein, nucleoprotein, and the last one is called as is also polymerase. That's also enzyme. There are two enzymes have been reported here. One is polymerase, otherwise called as DNA polymerase or RNA polymerase. Here is the RNA or DNA. The nucleic acid is the RNA. So RNA polymerase. Suppose if it is DNA polymerase. These are the types of protein also. Okay. See the different types. This is the structures. Paramyxovirus. Sometimes they may ask the question, N type of protein present the paramyxovirus. N denoted as which type? Which one of the following type of protein? Matrix protein or phosphoprotein or nucleoprotein or some other protein like the fusion protein like that. They may ask the question. So this is also an important one. This is a structure. Is just noted down the points where they may be indicated the protein molecules where there is a enzymes like that. You must note it down. Okay. See that rubella viruses. See here. This is the structure of rubella viruses. Simply we may call it as the outermost covering. There is a hook-like structure is called as glycoprotein, and the structure is called as similar to that of the what is a hexagonal shape. Sixth shape is the isohedral and nucleocapsid. Capsid is nothing but the protein covering. Inside there is a RNA, single standard positive sense. Single standard positive sense. Why it is called as positive sense? Why it is called as negative sense? Suppose if it is rotated in the right angle and also left angle. Likewise, they may be divided into a DNA molecule according to the rotation format. Okay. See that lipid bilayer membrane. Already you studied in the human plasma membrane structures, lipid bilayers. So this is the outermost covering. This is also lipid. This is also protein. So the both are present in it. That's why it is a very, what is that, resistant to many other antibiotics or antigens. Antibiotics. Okay, here given here, see that rubella virus are single stranded positive RNA. Single stranded positive RNA viruses. Diameter 50 to 70 nanometer. Nanometer is nothing but, will you please give me the unit of nanometer? Nanometer, nanoparticle.
So 10 power minus 9.
Good, very good. Exactly correct. Okay, well done. So envelope spherical in nature. See that structure look like envelope nature and spherical in nature. Spherical shape. Virus carry a hemoagglutinin. Virus multiply in the cytoplasm of the infected cells. Multiplication takes place in the cytoplasm. Okay, this is also a mechanism of viral rubella virus. That is also very, very important. Go to the next one. So the measles morbillovirus. This is also, the structure is not clear, but see here, it is also same. The phosphoprotein, lipid membrane, matrix protein, nucleoprotein, RNA is yellow in color. See here, this is the RNA structures. And this one is called as hemoagglutinin fusion protein. Just similar to that of the, which one you know? Which one you know? Sir, the paramyxovirus. Okay, paramyxovirus, rubella virus, and measles morbilloviruses. They are all are induced the, which one you know? Measles. Okay. See that. What are the symptoms? Already you know very but early symptoms are common cold. First induce the common cold. Next headache. It is also both are related. Headache as well as a cold, and also followed by fever, by coughing and sneezing, skin eruption, colored lesions called rubella conjunctivitis. Note it down. This is very, very important. This is a colored lesion. Okay. So the rubella virus is create the skin eruption as well as the colored lesions. That lesions also called as rubella conjunctivitis, etc. See the diagram, we can see it. This is the structure. See that small baby slide is here. The symptom of measles. Okay, see the dots. See the dots. This is a skins lesions. Okay, see the dry cough and runny nose, body pain and headache, sore throat, watering, swelling in eyes, and discomfort of fatigue, loss of appetite. You will not eat. Diarrhea continuously. What is that? Dysentery, diarrhea. Light sensitivity. Light sensitivity, cannot be able to see in light. Okay, it is just like what is that? You are string your eyes. Okay, inflammation in lymph node. This is a very complicated lymph node. Okay, and also last one is called as Koplik spot, blue red spot in the mouth. Blue red spot in the mouth also. Not only that, and also the body parts. See that there are small dot like stuff. These are all are concentrated. It is known as what is that? That symptom is called as what is that? Conjunctivitis, rubella conjunctivitis. That is called as colored lesions. Just like an inflammation, small dot are like structures have been analyzed, or a small spots also created on the human skin. Okay, see that. Prophylaxis. How will you? Okay, see that isolation of the infected babies. You have to isolate the person, otherwise called as what is that? Healthy person also. Okay, sometimes they may be move from the place to another one. That means you have to take care of the health. Isolation of the infected babies. And measles vaccine is given at 9 months, followed by the booster in the form of MMR. What is the meaning of MMR? Vaccine at 15 months of age. Listen here, how many years? 15 months. Yeah.
So MMR is measles, mumps, and rubella, right, sir?
Correct. Yes, correct. Exactly correct. That's why I'm asking. Sometimes they may ask the questions. How you know? What is the expansion of MMR? Okay. Already you know, already study. What is my BMR? What is my BMR? Have you heard about this word MMR? Already have heard. Yes, that's why you given an answer also. Okay. What about MMR? BMR, sorry, BMR. You don't know, isn't it? I will tell you. basal metabolic rate. BMR is nothing but the basal metabolic rate. Suppose your body metabolism is changed, due to the fever or something else. At that time, what happened? The basal metabolic rates also change. It can be measured as one of the equipments. Okay, just note it down. Okay. So first one is measles, and second one is we are passing to the next one is called as mumps. Okay, it is also one of the viral diseases, paramyxoviruses. That's why I have chosen the slide. Causes, it is caused by a RNA, paramyxoviruses. Mumps viruses, which generally attack children groups of 5 to 15 years. Okay, measles also 5 years up to. Here 5 to 15 years. Epidemiology, it is an acute contagious disease which is also spread by droplet infection. Both are same. Incubation period is here about 12 to 26 days. 1 month it will take, near 1 month. Okay, these are the, what is that? Important points. Epidemiology caused due to the which acute contagious disease and also by the droplet infections. And the incubation period is 12 to 26 days. See the paramyxoviridae, otherwise called as mumps virus. Here the outermost protein is also called as F protein. Okay, here Hudson protein. See that hook like needle-like structure is also present in it, in the outermost covering. Okay, and also M protein. Inside there is a covering is there, M protein. This is called as nucleic capsid. Nucleic capsid is covered by nucleic acid covered by a protein layer is called as nucleic capsid. The center there is a dark band-like structure, a oval-shaped structure is present in it. That is called as, what is that? Polymerase enzymes. See that a single stranded, it is not a double stranded. Double stranded RNA is not possible, but most of the cases single stranded RNA only presented. Some cases, exceptional cases are most 99% only the single stranded RNA will be possible. Double stranded RNA will be very, very, very rare. Okay, so this is very, very important thing. Mumps virus is also having the single stranded, negative single stranded RNA. That's why I have the symbol, negative symbol in the within the bracket. And these are the types of protein molecule. These are the 3 types of protein molecule, and also with the enzyme and also nucleic capsid is also one of the covering protein molecules. It is also made up of protein. Okay, see go to the next one. See symptoms. Mumps is characterized by painful enlargement of the parotid salivary gland. How many pair of salivary gland already studied? How many pair of salivary gland? 3 pair, isn't it? Sublingual. Yeah, good. 3 pairs: sublingual, submaxillary, and one more is that.
So parotid.
Yeah, parotid. So the parotid glands undergoes the enlargement due to the mumps. What happened? Your chin also swells, and sometimes other salivary gland also. Sometimes other salivary gland also affected. What is the main reasons? Symptoms due to the symptom of mumps is characterized by painful. It has a pain. Okay, enlargement of the parotid salivary gland and submaxillary and also sublingual. These are the 2 glands. Okay, and each it has 2 pairs. That's why 3 pairs of gland present, 3 pairs of salivary gland present in each and every human being. So also called as infectious parotids. The mumps also otherwise called as infectious parotids. Other symptoms are high fever, difficulty in the movement of jaws and opening of mouth, because it is swelling in the right side or left side. Okay, what happened? It will lead to what happening? You will not open the mouth. Okay, and because the pain is also present. See the next slide, you can easily understand. See that swelling of parotid gland. So the structure is swelling, isn't it? See the human face. Okay, so it's same side, it's also opposite side it may comes, because for your V is here so isn't it? That is also very, very important. Swollen of parotid glands, mumps due to the virus. Okay, so see that these are the symptoms. See the exact diagram. Okay, here this is the parotid glands. It's suppose if it is swell, what happened? So protrude one side, the chin also swells. and also pain, great pains comes out. See, prophylaxis, same things is there, similar to that of all the viral diseases, not only viral diseases and also other diseases also. You must isolation of the patient, like that you may told. Isolation of infected babies, mumps vaccine at 9 months to the baby. You have to provide 9 months. Okay, which one? The MMR vaccine. The MMR already the Ajay told the measles, mumps, and rubella. That's why the MMR is short form. It may be expanded in the measles, mumps, and also rubella. It's used against the 3 diseases: measles, mumps, and also German measles. One more is given. Okay, German measles. So this is also another type. So you just note it down. How many months you have to provide the under the diagnosis and also prophylaxis aspects, and also what are the vaccines? MMR vaccine. What is the expansion of MMR? Sometimes they may ask the questions. Which one you know? The match the following. Okay, we go to the next one. See the MMR vaccine, measles, mumps, and rubella. This is the structure. See that vaccine box also they mention, isn't it? See that. Okay. What are the next one? So this is also same. Rubella vaccine 20 ml. The content is also 20 ml. You have to give according to the age group. Suppose if it is the age group is more than that one, you have to provide more ml. Okay. So child or infant means according to the age group they will provide or they will prescribe by the doctors. Okay. See that. Next one is very dangerous one. Already known one. Rabies, otherwise called as hydrophobia. Phobia, nothing but fear, isn't it? Phobia is nothing but the very important factors that is called as fears. See that. When you bite any one of the rabies attacked by the dogs, isn't it? That is very, very important. The dog bite you, what happened, you know? Salivary glands contain numerous number of rabies viruses. The rabies viruses enter in your blood and also it reaches the brain directly. It is very, very complicated. See that one? The causes, it is a viral diseases caused by an RNA virus of the Lysa virus genus. Lysa virus genus. See here, this is the structures. Rabies, Lysa viruses. Epidemiology, see that human infection is indirect and inoculative. The viruses are injected in the human blood by the bite of rabbit animals such as dog, monkey, and cat, etc., along with their saliva. Already told you. Take any one of the animals like that, the dogs or monkey or cats or all, whatever it may be, the salivary glands present, so many microorganisms. Okay. Suppose if it's bite any persons, what happened in the salivary gland, the saliva also enter, the saliva contain numerous, what is that? Viruses. Which type of virus? The RNA containing Lysa viruses. It enter into the blood. After that reaching the blood, automatically it may be spread throughout the body, isn't it? That's the reason. When it reaches the brains, what happened? See that? Incubation period, it varies from 10 days to 1 to 3 months. Sometimes it may be happen up to 1 to 3 months. I think it may be 1 week or 2 weeks you will get the symptoms. Okay. So this is very, very important. They may ask the question, rabies also here, otherwise called as hydrophobia, and which type of virus and which are animals. Okay, what are the animals? They will spread the diseases when it spread. Okay, what are the incubation periods? This must note. These are the very important. See that structures. Here you must note it down. The rabies virus contain, these are the types of proteins. That's why here see that there are 5 types of protein also. M protein, N protein, G protein, L protein, and B protein. That was the nucleic acid RNA. The outermost covering is called as made up of protein molecules only. See that the entire structure is made up of which one in a protein type, different types of protein. Then, so what is the different types of protein? M protein, N protein, G protein. What is the meaning of that one? What is that? Proteins are made up of what is that? Which biomolecules? Ajay?
Sir, amino acid.
Sir,
Very good. Sir. Very good. Amino acid. How it will be form as a protein molecule? With the help of bond is there, isn't it? Which type of bond you know? So many bonds is there.
sir, diester.
No. Diaceto. No.
So polypeptide.
No. Two amino acids united together. What is that? Two glucose molecule united together with the help of which bond? Do you know the idea? Any idea about?
So peptide bond, sir.
Correct. So amino acids united with the peptide bond. What about glucose? Glucose means glycosidic bond. Phosphate bond means it may be differs. Okay. Okay. See here, these are the protein molecules. So how they may be differentiated, you know, because the combination of other molecule, N protein, M protein, also G protein. Glycoprotein means it's also connection with the protein molecule combined with the molecules of glucose or some other saccharide. That's why glycoprotein. Lipoprotein, lipid with protein. M protein is a mucus, or N protein is also amino protein. Okay, or some other component like that. Okay, anyhow, see that this is the structures. Matrix protein, phosphoprotein, phosphate also combined. G protein, that is called as glycoprotein. I already told you. Okay, M protein is nothing but the matrix protein. So one of the polymerase, which are polymerase is the RNA or DNA. RNA is the nucleic acid, so the RNA polymerase present. This is the another cross section inside. How it will be like that. So the slide. See that types of proteins in rabies virus already told you: nucleoprotein, phosphoprotein, matrix protein, glycoprotein, and also polymerase. Okay, the nucleoprotein is denoted as N protein, phosphoprotein, P protein, M protein, matrix protein, glycoprotein, G protein. Polymerase is nothing but the enzymes. It is also denoted as a lip L. Okay, so these are the type of protein mostly covered the which one you know rabies viruses. See that viruses also look like a structure. Okay, this is a electron microscope view. Okay, see that. See the structures of rabies symptom in dogs. Okay, see the salivary glands. Okay, salivary gland is very dangerous. See that dogs, it bites. It look like a very terrible in nature. Okay, see that. So this is the another slide. Okay, see when it bite the which one you know human beings, what will happen? When it bite the person, what happened? The salivary gland, okay, containing numerous viruses, they may enter into the blood. As a result, what happened in transfer from the one place to another place through the way of the connective tissues is called as what is that? Blood. Blood is a liquid connective. They can easily spread because it is also one of the nutritive medium, because virus they can grow only in the which medium? Will you please? Suppose take an example of COVID. Okay, when you are going to grow in the laboratory aspects, we are going to analyze or identify the diagnostic process, or you have to know the molecular structure of viruses. How will you grow? How will you culture? That is my question. How will you culture the virus in the lab? Which is the nutritive medium for their growth? Anybody else? Bacteria, how will you grow? With the help of the nutritive medium is the?
Yes, sir.
Which one? Agar? Ajay, sorry, already I told you answer. Agar, isn't it? Agar used most of the microbial growth, isn't it? Laboratory as well,
So
DMLT.
agar plate, sir.
Yeah, that's one for which one you know?
So bacteria, sir.
Bacteria. What about virus? I am asking. Virus only. How will you grow in virus?
Sir, don't know, sir.
You don't know. Anybody else? Dhanalakshmi? Okay, anyhow, I will tell you the virus only culture under the laboratory conditions, blood culture, blood. Okay, blood is one of the NEET medium because it's contain all the NEETs, and that's why any one of the diseases, viral diseases, can be easily spread through blood. See that. So these are the symptoms. What are the symptoms? See, the virus enter the tissue from the, which one, hitting of animals, biting or hitting of animals. Okay, this is the first symptom. And the virus replicates in the muscles near bite. Okay, so when there is a biting is take place here, the bite is taking place, isn't it? Here the surrounding there is a muscles is there. The muscles, what happen, the virus replication of DNA molecules undergo the replication process, and also multiplication takes place. It produces more numerous viruses. Then the virus move up peripheral nerve system to CNS. Peripheral nervous system is nothing but, only connection between the central nervous system and also the peripheral nervous system is nothing but the, what is that? A branch of the nerves. It may be spread throughout the body, isn't it? The central nervous system is the main, and also there are so many branches also started from the central nervous system. That is called as peripheral nervous system. And then virus ascend the spinal cord. Ascend means what happened here too? It may be enter into the spinal. Ascending, it may be move. Okay, into the, which one? No, another places. When it reaches the virus reaches the brain and causes the fatal and encephalitis. Fatal, sometimes it lead to death. Very dangerous one. Fatal is nothing but a very dangerous. Encephalitis, brain. Encephalitis is nothing but the brain. Okay, note it down. Where it happens. Encephalitis is nothing but the, sometimes there may be water or something as it happen inside the brain valley, storage of water, encephalitis. And then what happened? Virus enters salivary glands and other organs of the victims. These are the spreading pattern. Biting, replicating, move on to the peripheral, spinal cord and brain, and also salivary gland. Okay, because it is a very complicated. See that one? What are the symptoms? So already I told you, rabies is 100% fatal disease. Suppose if you are not taking any antibiotics, it will lead to death. Okay, within short period, because the virus is very, very dangerous, very dangerous. Okay, acute. What does harmful or fatal means? Causing death. See, fatal is nothing but the causing death. That's why I told you. Okay, it's a very, very complicated 100% of death. When you are not taking any one of the precaution, what will happen? It will lead to death. Rabies is characterized by the severe headache, high fever, and also painful spasmodic contraction of muscles of throat and chest. These are the very complicated. Already normal things and common cold or whatever it may be. These are the same things is that. But here, what happened? The muscles and throat and chest also having the pains. Not the muscles. It is not easily contract. So the patients feel restlessness, shocking, and difficulty in the, what is that? Woven liquid food. That is called as, what is that? Avoid, maximum they avoid the liquid food because it is called as, that symptom is called as hydrophobia, water hatting. Hydrophobia means nothing but, what is that? Fear about?
Fear of water.
Fear of water. Very good. Okay, fear of water. Suppose the rabies bite, what happened? These are the symptoms you are facing, isn't it? See that. He developed a fear of water, so called as hydrophobia. Severe damage to motor neurons of the CNS, paralysis and painful death. So these are the very, very complicated one. This is a very, very complex. That's why we have to aware of dogs. Okay. And prophylaxis, compulsory immunization of dogs. Dogs also immunized. Okay. And killing of rabbit animals. Particular animals also killed. Okay. Like that they told, but it is not possible. So many times you have to take care about of animals also. Suppose if it is dead, very, very dangerous. When it bites any one of the human bodies or human beings, within short period the animals are dead. What happened? You have to take very, very serious that matter. Okay, immediately you have to admit the hospital and also take the precaution. Then only it will be. Suppose if it is not like that, it will lead to some fatal. Okay, lead to death only. Okay. See that rabies vaccines, Rabipure. So these are the things. The rabies vaccine production, where it is it? Do you know? Where rabies vaccine is also produced in our Tamil Nadu. Which area do you know?
So no, sir.
You don't know. Okay. You know Ooty, isn't it? Ooty Kunnur, there is a Pasteur Institute. Pasteur Institute, where it located, you know, just note it down. Ooty Kunnur. But the rabies vaccine also production is there only, because the production need cold conditions. That's why Ooty is a favorable for the production of antibiotics, but such as the vaccine like that, rabies vaccine. Not only that, and so many vaccines also production takes place. Particular rabies is also one of the important one. That's why I have told. And next one is we pass on to trachoma. So another viral diseases, trachoma causes. Trachoma is caused by Chlamydia trachomatis. Chlamydia trachomatis. Just these are the genus, or these are the species name. Epidemiology, it is transmitted by the direct human contact contaminated object and by eye-seeking flies vectors. Eye-seeking flies. Okay, it's also one of the, which one you know, vectors. Eye-seeking flies. And also incubation period about 5 to 12 days. Okay, once again repeat for you. Trachoma is one of the eye diseases. Madras eye like that, you know, just like that, but it's not exactly like that. Trachoma is nothing but the eye disease. So cosmetic organism, Chlamydia trachomatis. How it will be transfer from the vector? That vector is called as, which one you know, eye-seeking flies. It's also one of the bees, like that. What will be the incubation period? Say about 5 to 12 days only. See that next one is, these are the Chlamydia trachomatis. This is the virus. See, just like a small oval shaped structures. When you are going to see the electron microscope, we can see the structure exactly oval in shape. Okay, just like a cherry, isn't it? Okay. See, these are the viruses connected to the OBC like that, or whatever it may be. See that life cycles of Chlamydia. Okay, Chlamydia is enter. This is the first stage division, and second stage it's also converted to multiplication. See that they mixed with the cell elementary body is EB and reticulate body, and finally it will due to some difficulties, lysis of rupture and cell membrane, because it's a virus, just like a lytic cell. When it enter, bind with the host cell, multiplication and rupture the cells, comes out, and again it will be entered into the same cycle. Already you studied in the lytic cycle and lysogenic cycles. Okay, like that. These are all no need. Symptoms. See, this is very, very important. Trachoma is characterized by the inflammation and discharge from the eyes, ulceration of conjunctiva and cornea, leading to blindness. Suppose if it is severe cases, lead to what happened now? Blindness. Before that, already in our, but trachoma is one of the eye defect, sorry, eye affecting diseases, isn't it? Trachoma is characterized by the inflammation and discharge from the eyes, ulceration of the conjunctiva and cornea, leading to blindness. It is one of the leading global causes of the blindness, and it is responsible for about 5% of blindness in India. Okay. Mostly those are having the eye defect, conjunctiva, ulceration of conjunctiva, or whatever it may be, leading to global cause of blindness due to this type of, which one you know, viruses. Only 5% total population of viruses, total population of India, the 5% of blindness is caused by the, which one you know, this type of viruses. And other symptoms also given here. Other important viral diseases of human are AIDS and HIV and hepatitis B viruses. AIDS already in our body. It is also one of the very dangerous viruses, acute immunodeficiency virus. It is, which is caused by the one of the dangerous viruses called as human immunodeficiency viruses, otherwise called as HIV viruses. So it's also having the glycoprotein outermost layer is called as glycoproteins. Okay, GP120 and GP41 only. The center there is one of the nucleic acid RNA containing viruses. RNA containing viruses, two single standard RNA molecule present in it, and with reverse transcriptase enzymes. Okay, how it will be enter? How it will be enter in our laboratory? When it enters, how it will be enters? Due to the sexual contact, and also blood transfusion, and affected person using the needles and syringe, that things also easily spread to the healthy person to, or affected person to healthy person. Okay, that is also very, very important things. And what will be inside, how it will be entered, it mostly bind with the, which one, beta lymphocytes and T lymphocytes. As a result, completely denaturation of the cells. As a result, what happens? They will not produce the T lymphocytes and the B lymphocytes. So immediately what happened, automatically, already about the immunity power will be completely lost. As a result, other diseases also enter inside. Particularly, the TP is very, very complicated. TP is a tuberculosis is the very fond of AIDS, very good friend of AIDS, like that you may tell. As a result, community immunity powers is also lost, so we cannot able to survive, isn't it? It lead to death finally. What are the symptoms? First of all, you note it down. When it enter, the symptom will be 1 month, 2 months, or 3 months. After up to 3 months, 6 months. Okay, then what will happen? Already about same as it is a fever, sweatiness, okay, and appetite, headache, and also the temperature suddenly increases, okay, and completely coughing, restlessness, body pain. These are all our concerned. At the same time, TP is very friend, already told you, very friend of AIDS, isn't it? TP is also one of the additional diseases. As a result, what happened? Completely loss of immunity. What happened? The human beings, they will not survive. So all the other diseases also, jaundice, that things also enter. So finally lead to that the body mass, okay, completely lose out. Okay. So how will you identify? There are the two things is that two method also identification is there. One of the main method is called as what is that? ELISA, enzyme linked immunosorbent assay. Okay. As a result, what happened? The ELISA is the whether the person suffering from AIDS or not, like that we can identify by this method. And another method is a confirmatory method. Confirmatory. The person must have AIDS. That is called as Western blot method. Okay. Another method also having one-dimensional, two-dimensional, one-step method, two-step method, and oral quick method. There are so many methods have been reported by, and there are so many vaccination also going on, but it is not exactly used for the AIDS. Okay. So nowadays the research also going on. 99% have been completed. Remaining 1% is there. After that, it will be comes to market. Okay. See the structure of trachoma. See, this is a symptom causes risk factor and also prevents. See that eye rashes. Inside there is a red color is there. This is due to the which one? The trachoma viruses. Okay. See, treatment of trachoma. The chlamydia trachomatis is sensitive to the tetracycline, deoxycycline, sulfonamide, and erythromycin. These are the four they have give sensitive. Organs lives inside the body cells, protect from full effect of antibiotics, respond to treatment therefore much slower. So you have to provide, these are the treatment for the trachoma diseases. Okay. So, and next common viral disease, already we saw it, these things fully. The poliomyelitis, okay, influenza, measles, chickenpox, it's a mumps, dengue fever, and also chikungunya. Okay, these are the things is also already we have to discuss. These are the common viral diseases mostly present in a book. Just to go and check it here. See that dengue viruses is not so one of the female arborovirus, always called as flavivirus. So Aedes aegypti, this is the causative organism. Okay, this is another one. And measles, influenza, orthomyxovirus, already we saw it, these things also. Chikungunya, dengue fever is also flavivirus. Okay, this is also another one. Okay, see that. So chikungunya, so dengue virus, flavivectors. See, Aedes aegypti. Okay, and rabies, already we saw it. Common cold, already we saw it. SARS, it is also another one. Sometimes they may ask the question, which one? Mnemonic aspect, the explanatory aspect. SARS, severe acute respiratory syndrome. Severe acute respiratory syndromes. Pneumonia-like symptoms. Respiration means already pneumonia. What are the symptoms? We already studied pneumonia. Okay. And also swine flu, already we saw it, H1N1 viruses. Okay, droplet infections. See that? So this is also common viral diseases in kettles. See that the skin also affected by this is the kettle. Okay, so many kettles also having viral diseases. These are all due to the RNA containing viruses. Common viral diseases in kettles. And foot and mouth also, sometimes they may happen. Okay, see that. See here, hepatitis virus is a very, very dangerous virus, similar to that of the, which one, oncogenic viruses. Here they have given is, which one, types of viruses, what are the symptoms, how it will be spread. See here, types, hepatitis A virus, B virus, C virus, D virus, and also E viruses. A to E viruses. Mode of transmission, see here, the fecal and oral, fecal matter. Hepatitis A virus. Hepatitis B, blood-borne sexual vesicle. This is hepatitis B also blood-borne, and blood-borne depends on HP, hepatitis B virus. Super or co-infection. These are the mode of transmission. Co-infection means nearest person suffered by things, and fecal and oral. These are the same. First one A and also E also same. Classification: picornovirus, viridae. Picornoviridae. Linear single standard RNA. See here, hepadinovirus, circular. Here the circular double standard DNA and RT enzyme, reverse transcriptase enzyme. That's why. And flaviviridae, linear single standard RNA. Delta viridae, circular single standard RNA. Calciviridae, linear single standard viridae. These are the types of RNA. See here, types. What are the nucleic acids? Incubation, see that all are similar. These three are similar. See here, 2 to 3 weeks, 2 to 6 months, 2 to 6 months, 60 to 180 days. How many days? 6 months means how much? 180 days. 21 to 42 days. These are the things. Chronic infection, no. Hepatitis B, yes, 10%. Here, 60 to 80%. Yes, no. This is a very complicated one. Okay, hepatitis C, hepatitis B also. Okay, and clinical outcomes of chronic infection, no here. And cirrhosis or hepatocellular carcinoma. This is very, very important. These two are very important. This hepatitis B and C is very dangerous. That's why I told you hepatitis is also similar to that of carcinogenic effect. Okay, cirrhosis or hepatocellular, and hepatocellular, what is that? Intestine, hepatic. Okay, tissues. Cirrhosis or hepatocellular carcinoma, both are same. Co-infection, same thing is there, and also no. These are the normal hepatic viruses also. Sometimes they may ask the hepatitis B viruses, particularly note it down. It is similar to that of the which one carcinoma effect. Most of them carcinoma effect means what is that? The sudden changes, sudden mutation to explain the nucleic acid and also spread throughout the body, throughout the body by the spreading. That is what is that? Spreading from the one place to another place. Okay, see that? See, these are the symptoms. Symptom: fever, headache, malaria, and jaundice. Severe liver damage, chronic disease occurs, same as HPV, more chronic, severe liver damage, high mortality rate. So pregnant women may at high risk at slow high mortality chronic diseases. 2 to 6 weeks, 3 to 26 weeks, 2 to 36 weeks, 6 to 26. These are all are just a incubation period. See the ingestion, parental sexual contact, hepatitis. See, this is very complicated. Okay, parental when the co-infected with HPV injection. These are the things. Hepatitis B virus only important things. And diagnostic test, which one is that? We have to analyze the immunoglobulin maximum. See immunoglobulin M only, immunoglobulin M, because it's a pentameric nature. Otherwise, it is one of the high molecular weight component. That is molecules, immunoglobulins. Immunoglobulin M antibiotics, here also immunoglobulin M antibiotic. PCR of viral RNA, PCR of viral RNA, immunoglobulin M antibodies here also same, immunoglobulin M antibody, PCR or viral RNA. So these are the things. Particular hepatitis B only, note it down. Immunoglobulin M, all are having same things, isn't it? So that is the things I think. See that hepatitis is nothing but the liver also. Okay, affected by the which you know there is viruses, hepatic B virus, A virus, C virus, D virus, and E virus also there. Okay, see that hepatitis viruses, liver particularly hepatitis. Okay, it's a very complicated, dangerous, lead to death only. Suppose we are not seeing what happened. Similar to that of the AIDS. Suppose hepatitis virus comes, okay, the person come, what happened? Definitely it has a death. No doubt at all. We cannot be able to cure. In primitive stage, we have to find out, then only we can easily move on to the diagnostic process. Okay. Suppose if it is not found earlier, what happened? It's very, very complicated. Most of the persons, most of the disease person, they may went for which one, 75%. After the 75% only, they are going to meet the doctor. They are going for the treatment process. Before that, they do not know, okay, how to identify the diseases. That is the reason. Viral diseases is very, very danger. Okay, so the common viral infection is that. That's all about that today. That means part 3 finish it. Okay, so not only finished. Okay, we are having another slides also on 4 and 5 and 6 also having. So we will pass on to the next part 3. So before that, we must summarize the lessons, summarize the things. The first one is what we discussed, measles, mumps, okay, and then rabies, okay, and then next what we did, what we discussed, rabies, and then which one, rabies virus is already trachoma is the last one, and isn't it? And also after that one, hepatitis finish means AIDS, and also Aedes, dengue. That things also you have to note it down, which are the vectors. That is a very, very important. Why the female is act as a vector, already told you, because the only the female it has a capability or ability to produce the egg only. Okay, others male it will not produce the egg. That is the reason. So the multiplication only by the female. So that is the reasons. Okay, that is very, very important things. Just note it down. Okay, so next we will pass on to the next one is called as what is the fourth one. Just I will give you an introduction, then remaining things will be on the next class. See, next the part 4, human welfare continue. The protozoan diseases. What is by protozoans? What are the diseases comes under these headings? That things we have to note it down. Protozoa is a minute microscopic organisms. Okay, see that. So these are the types of protozoa. These are the different types of protozoa have been reported, have been identified by the many scientists from the deceased or and water or whatever it may be, whether contaminated persons or specimens. That is condition is called as specimens, isolated from the affected persons, and also identified the structures according to their morphologically. There may be different types of protozoans have been reported by the scientists. Okay, see that protozoa and helminths, these things we are going to study. The flagellated, okay, see here, Giardia intestinalis, this type of which one, flagellated protozoa present the intestinals. Amoeba already in our body, it is also one of the protozoa. Okay, sporozoa, plasmodium already in our body. Sporozoa, it is produced the spores, then only it may be spread from the many things. Ciliates, see here, Palladium coil, these are the things, these are the protozoas. Not only this, and so many things is a plasmodium, already we discussed. Helminths, nematode, Asparagus lumbricoids is one of the nematodes. And termatode, okay, Fasciola hepatica, this is only present in the liver fluke, just imagine. Chestoda, Taenia solium, otherwise called as tapeworm. This is called as roundworm. This is called as tamatode, is a flatworm. This is cestode, is a Taenia solium, is a tapeworm. These are all are present. These are all are the parasite present in a human body. Okay, they are all are producing some diseases. Okay, that things we are going to discuss one by one. Yeah, see here, see. So this is the protozoan diseases. Already we discussed it. Bacterial diseases, viral diseases. Now we will pass on to the next one is called the protozoan diseases. And fourth one is called as helminth diseases. And then we will enter into the immunity. Immunity means you'll know where the fight against the diseases is called as immunity. The science, the deal study of the antigen antibody is called as immunology. Okay, you have to study that things also. They are divided into two categories. Okay, that things we will study in deeply. Okay, so now protozoan disease, what are the types? They are having so many types have been reported here, but we are going to study 4 or 5 only. Basic properties of protozoa. Protozoa, there is a basic properties. Each one it has a viral, it has a basic properties. Bacteria it also has one basic properties. Human beings also, plant also. Tetrabed, angiosperm, gymnosperm, okay, beta, whatever it may be. So and then amoebiasis already you know, amoeba is also one of the protozoa. Primary amoeba, meningioencephalitis. Encephalitis already told, brain related one. Okay, giardiasis, no need this one, just note it down. These are the present. Trichomonosis, Balandiasis, already we studied that. Toxoplasmosis, malaria, cryptosporidiosis, pneumocytosis. Here, malaria, trichoamoebiasis. These are the things we are going to study, particularly plasmodium. Okay, that things we will study the slides. First, amoebiasis only we will finish it today. So amoebiasis already one over. Amoeba is one of the, which one you know? Endoparasitic protozoa. Endo means it mostly present in the human intestine. Okay, only it lives within the cell only, within the living organism, within the host only. Causes, it is caused by the intestinal endoparasitic protozoan of human Endamoeba histolytica. Amoebiasis causes Endamoeba histolytica, found in the large intestine and lower part of the small intestine. Note it down. This is the very, very important one. The amoebiasis, the Endamoeba histolytica, mostly lives are found in the which part, like that they may ask the end of the small intestine and starting of the large intestines. That is very, very important. Epidemiology, infection is direct and oral. The tetranucleated cyst, note it down, or injected with contaminated food and water. Normally, the amoebiasis is mostly present in the food and water also. Suppose if it is a developed, what happened in a tetranucleated cyst conditions only, what happened, you know, enter in the human body with contaminated food and water. This is a contaminated meat, you know, about. Okay, water is contaminated by whatever it may. Suppose to take an example of a tank present in the where in our upstairs, isn't it? Suppose you will not clear or was it, what will happen in it? It will lead to some diseases like that, isn't it? Like that. So there is a reason. Symptoms here, the pathogen secrete the cytolysine. Note it down, the enzyme. Amoebiasis, okay, amoebiasis is caused by ulcer also. See here, cytolysine. Cyto means cytology. The science that deals with study of cell is called a cytology. Lysine means lysis. The cytolysine enzymes are secreted by the particular, what is that, microorganism, amoeba, Endamoeba histolytica. As a result, what happened, which damage the intestinal leading to the its ulceration. It's a damages the cells. As a result, what happened now? Ulceration comes out. Ulceration, you know, it's just with a small wound in the wall of the, which one, you know, the blood. Okay, resulting in the acute diarrhea and blood and mucus in the stools. This is the symptoms. You have to note it down here. 4 or 5 point is very, very important. Symptom: the pathogen secrete one of the enzyme is called a cytolysins, which damage the intestinal cells. So as a result, leading up the ulcerations. Final acute diarrhea. Okay, what happened in the blood and mucus in the stools. Patients also feel abdominal pain. Suppose those who are suffering from this amoebiasis, the abdominal pains also present. Abdominal pain. Okay, so it's a very hardest one. See that gastrointestinal amoebiasis by Endamoeba. This is the structure of amoeba. Okay, the amoeba enter. How it will be enter? See that the mature cyst. Okay, mature cyst means tetrad stage. Tetrad stage. This is very, very important. Tetrad stage enter into the intestine and also it create the ulceration and also produce the enzymes. The enzyme create the ulcers. As a result, what happens when the blood also and blood vessels also erode. As a result, it may be, that means particularly it erode first mucous membrane, then enter into the cells, then again enter into the blood. So as a result, the blood comes onto the where? Small intestine. Finally, it reaches the stomach. I mean, lead to the intestine. Finally, it reaches out in the form of mucus and stool with blood. Okay, see that this is the structure. Endomorph histolytica is one of the protoplasm. I mean, protozoa, ectoplasm, nucleus, endosomes also present. Ingested red blood cells, endoplasm, ingested bacteria, cell membrane, and pseudopodia. Pseudopodia mostly used for the false feet. They can move from the one place to another place. These are the things. See, this is the structures. See the tetrad cyst. They may ask the question sometimes. Amoebiasis, that means Entomoeba histolytica, which stage they may be entered in the body, like that. Cyst, that is heterod stage. See that 4 nucleus present, 4 nucleus. That condition is called a tetrad stage. We enter cyst, encystation. It may be converted to tropocytes. That condition is called as tropocytes. Again, encystation. Okay, see that. So again, it's encysted, and also, so this is the way to spread the Endomorph histolytica. As a result, what happened? The penis also comes up, cell membrane. See the structures. So this is another section. Cytolytic enzymes. These are the 2 types of enzymes. Cell is the, these are the enzymes. They can damage the cells. As a result, what happens? To induce a wound or produce the wound cells. Okay, so there is a prophylaxis, proper sanitation conditions, sanitary conditions. You have to wash properly. Proper coverage of eatables. Suppose if it is spread from the flies or something else, eatables should be very covered by the clothes or whatever it may be. To prevent the contamination of the food by the vectors, just the vector is nothing but the flies. Take an example, any vectors. Proper washing of fruits and vegetables before eating. Already I told you. So many vectors on the laying the egg on the fruits or vegetables. Okay, you have to wash it properly and then you use. Okay, otherwise it lead to diseases. That is the important things. See, so tomorrow we will see the remaining things. So today we started from the protozoan diseases, and before that we will finish it. Which one? Viral diseases. What are the viral diseases? Already we studied 10 viral diseases. Is there measles, mumps, okay, and also AIDS, okay, and the last one is called as a trachoma. That things also already we discussed. Finally, we will enter into the protozoal first diseases. We finish it. Which one? See that already we saw it. That things also Entomypha histolytica. You must note it up. This is also very, very important. So many times they were asked the questions. Okay. Thank you, my dear students. Thank you for the participation. Just to go through it, you will get it after finishing your exam. You give full effort to this one. Okay. Thank you. Bye. Take care.
Thank you, sir.
Thank you, Kana. Thank you, ma'am.
Thank you, sir.
Thank you, ma'am.